Hello dear learners, welcome to the session. Today we are going to discuss the Watson and Crick model of DNA structures. So, perhaps most fundamental property of all living organisms is the ability to reproduce. All organisms inherit the genetic informations of specifying their structure, functions from their parents. Likewise, all cells arises from the pre-existing cells. So, the genetic material must be replicated and passed from parents to a progeny cells by the cell divisions. The mechanism of genetic transmissions and identification of the genetic material as the DNA was discovered that formed the fundamentals of our current understanding of the biology at molecular level. This DNA, initially the structure nobody has been elucidated. And Rosland obtained the X-ray diffraction photographs of DNA fibers. Different types of the DNA fibers they have been recorded. They have taken a photographs. The one which was uninterested, that has been this diffraction patterns demonstrated that DNA molecules how was helical shapes. James Watson and Francis Crick informed the characteristics of the DNA structures from these patterns in 1953, which are now widely accepted as the structure of the DNA. As you have seen in the X-ray crystallography photograph, this crystallographic analysis of the DNA in a nutshells, which consisting in a 1 turn 10 base pairs. The distance arises from 3.4 angstrom unit for each basis. The distance from the center is 3.4 angstrom unit. Twist from one point to another point, that is 360 degree circle, it will be 34 angstrom unit. This is in a nutshell. Let us look into this one in detail. What Watson and Crick elucidated, 4 point has been postulated by analyzing this photograph. The DNA is composed of complementary strands, that 2 strands will be there. These 2 strands will be twisted together among the same axis. At the central axis, this DNA has been twisted and forms the helical structures. These strands are moving in opposite directions. One strand moves from the 5 prime to 3 prime, another strand move from 3 prime to 5 prime. The outermost, this one will be polynucleotide chain will be there. This has been held together. In opposition to one another, these 2 polynucleotide chains are oriented. Helix is formed by the sugar and the phosphate group that makes the backbone of the DNA molecules, which nitrogenous base which will be projecting inwards, as seen in the picture. The running will be the backbones, the continuous structure, this one. The inside in a colored, this one which one we are showing, that is in a nitrogenous base. Nitrogenous base is a nonpolar and hydrophobic in nature, whereas the backbone chain is in hydrophilic, which will be projecting an outside of this one. As a simple words, these axes, so this DNA will be like a ladder like structures. This ladder like structure you can see in the first left photograph, this one, first line will be ladder will be from 5 prime to 3 prime, another one is 3 prime to 5 prime. The nitrogenous base lies perpendicular to the common axis. Suppose in the common axis, the nitrogenous base will be there. It is exactly at 90 degree, particularly perpendicularly it will be lies in this one. These bases are stacking one upon the another. This distance between the one base to another base is 3.4 angstrom unit. These bases are, how many bases will be present in this one means it will be 34 angstrom unit, 10 bases will be there, one turn to another turn, complete 360 degree. So, this one the length will be 34 angstrom unit. In this 34 angstrom unit, 10 bases will be accumulated. The one circle means it will be a 360 degree, 10 if you are divided by, each bases will be inclined to a 30 degree this one. Suppose this is a one base, is another base. If you are looking in this angle will be exactly 36° it has been twisted. If you look into the length of the diameter of the double helical structure as per the X-ray crystallography, it will be 20 angstrom unit. These two strands, how these two strands are held together? These two strands are held together by the hydrogen bond between the nitrogen bases, purine and pyrimidines will be there. Adenine always pairs with the thymine, guanine always pairs with the cytosine. The specific pairing is known as the base pairing. It allows two strands to fit together in a complementary way. You might have seen in the look in the diagram where cytosine and guanine 3 hydrogen bonds will be there, thymine and adenine will be 2 hydrogen bonds will be there. This specific pairing is the double helical is held together by one is hydrogen bond, it has been held. How this base pair having a distance of 3.4 angstrom unit? This is mainly because of the van der Waals force. This van der Waals force make them to stacking perpendicular each other. And these nitrogenous base are hydrophobic in nature, that is the reason they are tend to present in the inner side, whereas hydrophilic backbone are present on the outer side, because in a cell, in a nucleus, it will be relies on the aqueous environment. That is made to show as in a double helical structures. Thank you.